Hello, everybody. It's a pleasure to speak at this year's Genomics Live conference. I have to say that I was actually really excited to be part of the real event, which brings together so many interesting people right in the Pharma and Life Science Hub in Basel. Nevertheless, I'm now looking forward to the virtual conference sessions. In the next 20 minutes, I will introduce you to some of the technologies that we have developed within CELLINK, and I will also run you through a couple of workflows that we established together with partners and customers to analyze single cells from clinical samples. This is an overview of how I structured the presentation for today. After a short introduction of our company, I will introduce you to our technologies for single cell isolation and low volume reagent dispensing. Then I will present three different workflows. First, a workflow for full-length transcript sequencing from single cells, then a workflow for RNA sequencing of rare cells obtained from liquid biopsies, and finally, a solution for analyzing copy number variations and the mutational landscape in thousands of individual cells obtained from clinical samples. Let me start with a very brief introduction of our company. CYTENA is actually based in Freiburg, Germany, which is just less than an hour drive from Basel. Since last year, CYTENA is part of the CELLINK Corporation, a young company that has grown very rapidly over the last years. Founded in 2016, CELLINK is now present in over 60 countries, and we have offices pretty much all over the globe to develop new products and support our customers. Very recently, we had the pleasure to welcome two more sister companies, the world leader in precision dispensing Scienion and Cellenion, a company that has developed a great platform for single cell isolation and omics analysis. Within CELLINK, we are able to offer a growing portfolio of tools to enable efficient workflows throughout the life sciences, ranging from 3D bioprinters to low volume liquid handling instruments and instruments for single cell isolation and analysis. So how does single cell dispensing works? This is a schematic. The core is always a liquid dispenser that allows to generate tiny droplets. The nozzle of this dispenser needs to be transparent, such that a camera can be used to visualize, detect, and analyze cells in the nozzle of the dispenser. Using image processing algorithms, this allows to determine whether a single cell with a specific morphological and optical properties is going to be dispensed with a droplet. Finally, one needs a system to remove droplets that are either empty or contain multiple cells. This can be done by removing the droplets with vacuum suction before they can reach a target substrate, like a microwell plate. Another strategy is to dispense unwanted droplets into a waste reservoir, which requires to move back and forth between a waste position and target microwell. While the first strategy is typically faster, the second approach allows to recover cells that were dispensed into waste, which can be beneficial, for example, in rare cell applications. As you will see throughout my talk, there are several benefits over droplet-based technologies which are widely used for single-cell RNA sequencing. Single-cell dispensing is compatible with low sample volumes. There's almost zero cell loss, as I will show later. It's compatible with all common cell types, including nuclei, neurons, cardiomyocytes, and even small cells such as bacteria. It's compatible with a wide range of different assays. This makes the technology ideal for clinical samples and liquid biopsies. We and our sister companies at CELLINK have developed a variety of cutting-edge picoliter and nanoliter dispensing technologies. They all have in common that they are drop-on-demand technologies which offer precise control over drop volume, droplet stability, and dispensing frequency. They differ in the droplet generation principle, which ranges from direct displacement to acoustic wave, and the application of controlled pressure pulses to a liquid reservoir. Based on these technologies, we have developed a variety of laboratory devices. The Xcyte series and the CELLEN1 are instruments for single-cell dispensing, while the I.DOT and the I.DOT Mini are instruments for low volume reagent dispensing, for example, for cost-efficient and automated NGS library preparation. Before I will run you through some of the application examples, I will provide a short introduction to the three technologies. Let's start with the X-side instruments. This here is the F-side, which is a benchtop single-cell dispensing instrument that comes with a very intuitive software and is very easy to use. If we zoom into the printhead here, you can see the nozzle of our disposable dispensing cartridge. We use an optical detection system similar to a bright field microscope to detect cells in the nozzle of our dispensing cartridges. And only those droplets that contain a cell with the right properties are then deposited onto the target substrate. You can also see one of our single-use cartridges. Before single-cell dispensing, the sample is simply pipetted into the reservoir of one of the cartridge, which come sterile packaged, and they can hold between 5 and 80 microliter of sample. In fact, the cartridge is the only part that gets into contact with the sample, so there's no cleaning procedures required, and also there's no risk for cross-contamination between samples. For each cell that is dispensed, several images are stored to the hard drive to document the cell isolation process. Once the fluorescent laser is switched on, additional images from the fluorescent detection channel are stored along with these bright field images. As you can see on this slide, the F-side is compatible with a wide range of different cell types, including primary cells such as hepatocytes, neurons, and cells collected from blood samples. The CELLEN-1 is a highly flexible solution for single-cell and reagent dispensing, which is especially useful for ultra-low volume applications and rare cell samples. In contrast to the X-side instruments that I just introduced, here the sample can be aspirated from a source well or an Eppendorf tube into a glass capillary. Further, droplets without single cells are not removed into waste, but are dispensed into a dedicated tube from which they can be aspirated again if needed. The instrument comes with up to 4 fluorescent detection channels to sort based on cell viability or target subpopulations. For lysis and amplification in ultra-low volumes, the instruments can be equipped with a second reagent dispenser that allows to distribute reagents in the picoliter and nanoliter range. And we also offer a wide range of add-on modules to enable customized protocols and low-volume applications. Now let me also briefly introduce you to the I.DOT, which is a benchtop instrument for low-volume reagent dispensing. The I.DOT technology allows to generate droplets using a well-controlled pressure pulse that is applied to a liquid well with a small hole on the bottom, which you can see in the schematic on the lower right side. The I.DOT comes with 8 of such channels, which can generate droplets with volumes between 8 and 50 nanoliters, and volumes in the microliter range can be easily addressed given that droplets are generated with frequencies of 100 Hertz. Something that really sets the I.DOT apart from other low volume liquid handlers is the dispensing source plate, which holds up to 96 different reagents. It basically looks like a normal 96 well plate that can be filled with standard pipettes and can be moved around by ordinary plate handling robots. The system can dispense from any of these 96 wells into any well of a SBS format target plate, which allows to perform even the most complex liquid handling tasks within minutes. This summer, we have launched the I.DOT Mini, which is an extremely fast and affordable small brother of the I.DOT. So now in the second part of my talk, I will guide you through 3 application examples. Due to the limited time here, I will introduce each example only briefly. Please feel free to contact me after the talk if you want to discuss the workflows in greater detail. The first example I want to highlight is a method for full-length transcript sequencing from single cells. Here we established a highly cost-efficient protocol for SMART-SAC2, which is a popular and one of the most sensitive assays for single-cell RNA sequencing. In contrast to some other methods for single-cell RNA sequencing, this assay has to be performed in microwell plates, as it requires to generate cDNA libraries before the cells can be barcoded and pooled for sequencing. Here's an overview of how we set up the workflow. For the validation study I will present you today, we used a HEX cell line. The cells were diluted to 0.8 million cells per milliliter prior cell isolation, and then they were isolated with the F-side into 384 well plates pre-filled with lysis buffer, with 1 microliter of lysis buffer. And for the first part of this protocol, we used a commercial version of the SmartSeq2 assay from Takara to generate the cDNA libraries. And for the second part, the NGS library preparation, we used the Illumina Nextera XT kit. All reagents were dispensed automatically using the IDOT and 1 IDOT pure plate. All reactions were performed at 10-fold reduced volumes, which resulted in a significant cost reduction of these 2 most expensive parts of this protocol. This slide highlights the kind of data you can get from each individual step. As I mentioned earlier, we obtain images of individual cells that are isolated with the F-side. Here are some examples of HEX cells that have been dispensed. The F-side also dispenses empty droplets from the cell sample for a negative control if needed, and this is depicted in the 4th image here. After reverse transcription, the cDNA is amplified, resulting in cDNA libraries from single cells as depicted in the center, segmentation, and another round of PCR to bring in the sequencing adapters, then finally results in the sequencing library, which can be seen on the right. And these libraries can be then pooled into a single tube for sequencing. One thing that you can do with the images that we obtain from the F-side is to link them to the data generated throughout the assay or the final sequencing data. Here we looked at the cell size as determined by the F-side versus the cDNA yield after the amplification reaction. It's quite nice to see that there's indeed a good correlation between cell size and transcript abundance. For the analysis of more heterogeneous samples, this might be a helpful tool to improve normalization or clustering. Finally, we pooled 125 cells and sequenced them on an Illumina NextSeq instrument. As you can see here, we obtained high-quality sequencing data with a mapping rate of over 94%, and we detected almost 10,000 genes on average at 1 million reads. Since the entire protocol is based on 384 well plates, this setup should allow for the preparation of at least 1,000 to 2,000 single-cell libraries on a single day. Let me show you this slide to highlight the uniqueness of the IDOT for such workflows, and especially for efficient preparation of the NGS libraries. This is a screenshot of the Assay Studio, the software that comes along with the IDOT. It shows an example for barcoding 96 libraries. On the left, you see the source plate loaded with 12 forward and 8 reverse primers from the Illumina kit. And on the right, you see the 384 well plate, the target plate with the 96 cDNA segmented libraries that will be indexed. A nice feature of the IDOT Assay Studio is that it allows you to run a simulation prior you actually start the dispensing run. And as shown here, the time needed to index all these 96 libraries is just a minute. Given that up to 96 primers can be loaded onto the IDOT source plate, one can barcode more than 2,300 libraries at once. Obviously, any cross-contamination between the primers has to be avoided, since this would make proper multiplexing impossible. And this cannot be achieved with other single or multi-channel low-volume dispensing instruments that are not capable of non-contact dispensing all these barcoded primers at the same time. Now we come to the second application that I want to introduce today. This is an example of a quite demanding single-cell application that is addressed by a team at the UCSF in San Francisco. Their goal was to establish a protocol for isolating and sequencing cells from cerebrospinal fluid. To do so, they first draw 20 milliliters of cerebrospinal fluid from patients. And after separation and centrifugation, we end up with 5 microliter sample containing between 20 and 500 target cells. The biggest challenge for this and similar scenarios is that it's super difficult to obtain single cells from such minute amounts of sample. Due to high cell loss and the need for larger volumes, it's impossible to isolate these cells with FACS instrument or other existing technologies. As I will show you on the next slide, the aspirate and dispense capabilities of the CELN1 enable to process such samples with almost zero cell loss. After single cell isolation, the cells can be processed in microwell plates using SmartSeq2 or other methods for single cell RNA sequencing. As it is crucial for such workflows to minimize cell loss, we quantified the cell loss on the CELN1 with stained PBMCs from blood samples. From 3 such samples, we prepared 5 microliter aliquots, of which some were used for manual cell counting on a fluorescent microscope, while 1 aliquot was used to isolate cells with the CELN1. By manual counting, we obtained on average 68 cells, while after single cell dispensing, we counted 64 isolated cells. We did this with 3 different samples, and as you can see, almost all cells present in the sample could be isolated for subsequent analysis with the CELN1. The third example I want to highlight is a really powerful method for analyzing the genomes of thousands of single cells obtained from patient samples. It's a method called Direct Library Preparation Plus, which was developed at the University of British Columbia in Vancouver and the BC Cancer Center. The entire library preparation workflow was established on a CELN1 device, and the results from sequencing over 50,000 single cells and a detailed protocol were published last year in the journal Cell. Unfortunately, I do not have the time to go into a lot of detail here. For those of you that are interested in this detailed protocol and the results, I recommend to have a look at the free access paper. Here I will demonstrate the general idea and how it can be addressed with the CELN1. On the top, you see the overall concept of the library preparation part. From a heterogeneous cell population, such as dissociated tumor tissue, cells or nuclei are dispensed into nanowells for subsequent lysis. Then the DNA is directly segmented before the fragments are amplified by a minimal PCR, which also brings in the barcodes for sequencing. Finally, up to 5,000 cells are pooled via centrifugation for subsequent sequencing. Since thousands of cells are sequenced together, the coverage per cell is typically relatively low, depending on how much you sequence. However, the bias introduced during the process is extremely low due to the minimal PCR amplification. This enables large-scale copy number variation analysis on the single cell level, and after clustering and genome pooling, even single nucleotide variations as well as the mutational landscape can be obtained from this data. In the original publication, you will find extensive data sets on different samples, including nuclei, cryopreserved samples, dissociated tumor samples, and patient-derived xenografts. In total, cells ranging from 5 to 80 micrometer in size were successfully analyzed, and with 27 US cents per single cell library, the cost is extremely low, which makes this method suitable for routine applications. In fact, the British Columbia Cancer Center Research Center has already sequenced over 400,000 cells. Dead cells can be excluded from the library preparation based on fluorescent markers, and the authors of the paper also show that the images generated can be correlated with genomic properties. The protocol utilizes a commercial nanowell plate, which allows to barcode and pool up to 5,000 cells. The group has not only established a very robust protocol, but also developed an extensive data analysis platform. Therefore, several leading institutes have already started to establish this protocol in their labs to analyze clinical samples. With that, I'm almost at the end of my presentation. I hope that I was able to demonstrate the usefulness of single-cell dispensing technology for single-cell omics analysis of clinical samples. Let me finish with a short summary of the key features. Single-cell dispensing is compatible with a wide range of different cell types, including difficult to work with samples such as neurons, cardiomyocytes, or nuclei. The combination of almost zero cell loss and fluorescent sorting capability makes the systems ideal for liquid biopsies and rare cell samples, such as cerebrospinal fluid, as I demonstrated, or circulating tumor cells. In addition, the systems are compatible with a wide range of different substrates and assays, including emerging single-cell multi-omics methods. I also introduced you to the I.DOT, which we used to generate sequencing libraries. The I.DOT is one of the most versatile low-volume liquid handlers on the market. It features a low dead volume of below 1 microliter. It allows to load and dispense up to 96 reagents in parallel, and it also can dispense reagents loaded with glycerol or PEG, which are sometimes used as molecular crowding agents, for example, in the new SmartSig3 protocol. Let me also mention that the I.DOT is faster compared to comparable acoustic dispensing technologies. We just launched the I.DOT Mini this summer, which is super easy to use, and it comes at a fraction of the price of any other low volume dispensing solutions. If you are interested in our technologies or in discussing how we can enable your application, don't hesitate to contact me directly or our team at info@cytena.com. We are happy to schedule individual telecons, and our great team of application scientists in Freiburg and Lyon is capable of helping you to establish efficient and robust single-cell analysis workflows. With that, I'm at the end of my presentation. I hope you enjoyed it. Stay safe, enjoy the conference, and with that, I'm also happy to address your questions.